Welcome to a walkthrough of WEGAN's ordination tools for analyzing ecology data. WEGAN is a web-based platform for analyzing ecology data that requires no coding knowledge. In this step-by-step tutorial, we will show users how to use ordination analysis tools to perform multivariate plotting techniques such as PCA, NDMS, and PCOA. Let's get started.
Start by clicking the Ordination module to explore constrained and unconstrained ordination analysis methods. Next, upload your dataset using the Choose File button. Alternatively, users can select a sample dataset. For this example, we will select the dune sample dataset on grass species in a meadow. Selecting the dune dataset will also load the accompanying environmental and taxonomic data. Click Submit to proceed with your chosen data file. The Data Integrity Check page will confirm whether the data is usable, such as checking for correct sample and variable labeling. See our data uploading tutorial for more details on how to upload data and manage errors. Here we see the dataset has passed the integrity check. Scroll down to see a PCA plot of your data. Switch to the Heat Map tab to see a heat map of your data. The Data Editor tab can be used to make slight adjustments to the data. Here we can change the data type, edit the data, and remove entries. Find more information in our data uploading tutorial. Once ready to continue, scroll down and click Proceed. On the normalization page, you can select the normalization methods appropriate for your dataset.
There are 4 manual data transformation options to choose from. Click a radio button to select a methodology. Select the appropriate parameters and then click on the manual button. A pop-up will show you variable data distribution plots comparing the effect of your selections. In this example, no normalization was applied. To make revisions to the normalization, click Go Back. Going back, we can alternatively choose automatic normalization to apply recommended normalization settings based on Pearson mode skewness.
To move on from the normalization step, click Proceed.
For ease of replication, this tutorial will continue with no normalizations.
The second Data Integrity Check page verifies data quality in the supplementary data. This page is checking the environmental data. We see our data has passed the integrity check. Click Proceed to continue. The third Data Integrity page is for the supplementary taxonomic data. We see our data passed the integrity check. After completing data preprocessing, you can select the type of ordination analysis to perform. For this example, we will select canonical correspondence analysis. We have now reached the main analysis page. Here we can adjust our plot parameters and view our results files. Canonical correspondence analysis is an asymmetric technique of illustrating unimodal species abundance distributions along the environmental gradient. 2D ordination visually represents relationships among samples or variables in reduced dimensional space, revealing patterns, gradients, or clustering in complex data. Select whether to apply relative abundance transformation to your data, where each value is divided by the sum of the species variables. You can also select to use original non-normalized data. Next, select which constraining variables to use. Click Update to apply your changes and regenerate the plot. Scroll down to view your CCA plot. Click on Results to open a list of data files to download. Here we see multiple downloadable data files, including the summary data, ANOVA data, sample and variable score data, and constraining variable score data. Click on the desired file to automatically download. You can modify the appearance of the plot by clicking the Customize Graph button. Start by selecting plot display features such as labels, arrows, and ellipses. Next, adjust what variable to group the data by. Finally, select a color palette. Click Update to apply your changes to the plot. Scroll down to view the customized plot. Click Export to download this plot. Select the export format from a drop-down menu. We can select from PNG, TIFF, PDF, SVG, and PostScript formats. Next, select your desired export resolution. Options range from 72 dpi to 600 dpi. Finally, select export size of either full page or half page. Click Submit to generate the export file. The download link will now update. Click it to open the export file in a new tab. The Ordination module also produces scree plots. Click on the scree plot tab at the top of the page. The scree plot calculates what proportion of variance is explained by each dimension using eigenvalues. Adjust the variables as before. For this plot, we can also select the plot type. The classic plot displays eigenvalues. The variance explained plot shows both accumulated variance as bars and individual dimension variance as a line. Click Update to apply changes. Scroll down to see your plot. In this variance explained plot, the bars represent accumulated variance while the line represents individual dimension variance. Export this plot as before. Moving on, click on the RDA submodule from the navigation tree on the left. Redundancy analysis, or RDA, constrains community data to be linearly related to a second set of variables so that it can be plotted in a reduced space. It depicts linear distribution of species abundances along the environmental gradient based on the Euclidean distance measure. First, select whether to apply relative abundance transformations, where each value is divided by the sum of the species variable. Only apply this if your data contains samples and species abundances. Next, select the constraining variables. Multiple can be selected. Update to apply your changes. Click to customize the graph. Multiple plot display options can be selected to customize arrows, centroids, ellipses, and grouping options. The grouping variable can also be selected here. Select a color palette. Click Update to apply your changes. Scroll down to view your customized RDA plot. This plot displays the species linearly and the grouping category, moisture, as data points. Remember to export your plot. View all the relevant data files under the Results banner. Switch over to the scree plot tab. We will now apply the scree plot to our RDA to calculate what proportion of variance is explained by each dimension using eigenvalues. First, select whether to apply relative abundance transformation. Next, select the plot type. Recall that the classic scree plot displays eigenvalues, while the variance explained plot shows both accumulated variance as bars and individual dimension variance as a line. Select whether to display percent labels. Finally, select a line color. Click Update to apply your changes. Scroll down to view the scree plot. Download the data files related to this plot under the Results banner. Continue to the CA submodule. Correspondence analysis allows for a visual, Euclidean interpretation of frequencies of categorical variable combinations, commonly depicted as rows, sites, and columns, species, of a contingency table. In community composition data, the abundance values are the frequencies. Variable values can be represented as scores in a new orthogonal dimension. These sets of scores represent both rows and columns on the same scale, allowing the relationship between different variables to be examined. Note that CA considers unimodal species responses and relative abundances using the chi-squared distance symmetrically for sites and species. First, select whether to use non-normalized data. Next, select from the name and arrow options for your plot. Finally, select whether to use colors in your plot. This will turn the variable species data points red and the sample site data points blue. Click Update to apply your changes. Scroll down to view the plot. The biplot displays relationships between categorical variables. Points closer together indicate stronger relationships. In this plot, the variables are displayed as plus, and the samples are displayed as circles. Click Export to download your plot. Download the CA analysis results under the Results banner. Continue to the scree-plot tab. Adjust the parameters and update the scree-plot as before. Scroll down to view the scree-plot. In this version, percent labels are displayed. Download related data files and export the plot as needed. Continue to the NMDS submodule. Non-metric multidimensional scaling analysis, or NMDS, iteratively repositions points until the relative distances in the ordination have the strongest possible monotonic fit to the pairwise dissimilarities between sites. The NMDS 2D plot arranges points based on dissimilarities, aiming to maintain relationships between distances. Similar samples cluster closely. Select whether to use non-normalized data and whether to apply relative abundance transformation. Next, select to display icons, points, or text for either the sites scores or for species scores. Sites scores can be points, text, or icons. If not using sites scores, select None. Species scores can be points or icons. If not using species scores, select None. As NMDS applies random processes to generate valid analyses, the randomness setting can be adjusted. Select New Random Seed to perform a new analysis, or select Use Current Seed to maintain current settings so that plots are comparable. Finally, select a distance metric for calculating dissimilarity. Click Update to apply your changes. Next, click on Customize Graph. Select a grouping variable if desired. Adjust the plot display settings to include ellipses, variable arrows, or variable centroids. As this is a sites scores plot, we can adjust the color setting for the sites. Input a legend title if desired. Adjust the site display, margin, and text sizes as needed. Update the plot. Note that when Display as Icons is selected for the Sites Scores plotting parameter, additional customization options appear. Here, users can adjust the icon size and input a legend title. Scroll down to view the customized NMDS plot. In this plot, similar samples cluster closely. The arrow is displayed due to the customization setting, Display Environmental Variable Arrows. Download this plot using the Export button. Download the data files related to this NMDS plot under the Results banner. Switch over to the scree-plot tab. The NMDS scree plot shows the stress for NMDS solutions calculated with increasing dimensions. Stress is a measure of goodness of fit of the ordination. The stress function measures the difference in pairwise similarities between the data points in the original high-dimensional space versus the projected low-dimensional space. Adjust the parameters as needed. Select whether to display stress value labels. Click to update the plot. Continue to customize graph. Select a plot theme. Select whether to display grid lines. Adjust the text size. Update to apply your changes. Scroll down to view the NMDS stress plot. The red line shows the variance explained by individual dimensions. An inflection point on the graph can be used to help determine the optimal number of dimensions, but note that increasing dimensions impact interpretability. Download the data files and export the plot as needed. Continue to the Shepherd Plot tab. A Shepherd plot compares the original pairwise dissimilarity between data points against the ordinated dissimilarity distance calculated by NMDS. Adjust the plot parameters as needed. Select the number of dimensions to assess. Click Update to apply changes. Click to customize the graph. Adjust the theme, select whether to display grid lines, and adjust text size as needed. Update to apply changes. Scroll down to view the customized shepherd plot. If the NMDS was successful in representing the data in lower dimensions, data points will swarm around the line on the plot. Download the data files under the results banner and click Export to save the plot. Proceed to the PCA submodule. Principal Component Analysis, or PCA, generates linear combinations of the original variables, principal components, which maximize the variance. Pairwise dissimilarity between data points is measured using Euclidean distance. Select the number of principal components to apply. Select a color palette. Finally, select a grouping variable. Update to apply your changes. Scroll down to view the PCA plot. This pairs plot presents scatter plots of pairs of PCs, helping identify patterns and correlations in reduced dimensions. Click the Export button to save the plot. Download the data files related to this plot under the Results banner. Continue to the 2D Scores Plot tab. We can manually select two PCs for this plot. Select a grouping variable if desired. Select which principal component to plot along the x-axis. Next, select which principal component to plot along the y-axis. Select a color palette. Update to apply your changes. Click to customize the graph. The plot options include displaying arrows, ellipses, and sample names, and add grid lines. Click Update to apply changes. Scroll down to view the customized PCA plot. Click Export to save the plot. Download the analysis results related to this plot under the Results banner. Switch over to the Loadings Plot tab. The Loadings plot displays the loadings, which are the contributions of variables to the principal components. This is in contrast to the Scores plot, which shows the scores of the PCs. The scores apply to the samples, and the scores for each PC are the coordinates used to make the plot. Select the x-axis and y-axis PCs. Select whether or not to label the variables. Click Update to apply your changes. Scroll down to view the customized loadings plot. Download the data files and export the plot as needed. Continue to the Scree Plot tab. For this Scree plot, we only need to select the number of PCs to assess. Click Update to apply your changes. Scroll down to view the Scree plot. Download the related data files and export the plot as needed. Moving on, click on the PCOA submodule. Principal Coordinate Analysis, or PCOA, represents the pairwise dissimilarity between data points using a specified distance metric. Start by selecting the plot data options. Select whether to use relative abundance transformation, check for data presence or absence, or use original non-normalized data. Select a methodology for calculating distance measures. Select the constraining variables. Update to apply your changes. Next, customize the graph. Adjust the plot display options, such as displaying ellipses, arrows, centroids, and sample names. Adjust the grouping variable and color palette. Update to apply your changes. Scroll down to view the customized PCOA plot. The more similar their variable values, the closer the samples are to one another. Download the results files and export the plot as needed. Switch over to the Scree Plot tab. Adjust the Scree Plot parameters. Click Update to apply your changes. Scroll down to view the updated Scree Plot. The green line on top shows the accumulated variance explained. The blue line underneath shows the variance explained by individual dimensions. Download the results files and export the plot as needed. Continue to the Shepherd Plot tab. The Shepherd Plot parameters are as before. Adjust as needed. Update to apply your changes. Scroll down to view the plot. If the PCOA was successful in representing the data in lower dimensions, data points will swarm around the red line on the plot. Download the data files and export the plot as needed. Finally, move on to the polar Bray-Curtis submodule. Bray-Curtis ordination, or polar ordination, uses a distance metric to arrange samples between two endpoints, or poles. The first endpoint has the highest variance of distances to the other points. The second endpoint has the lowest regression coefficient between distance from the first and second point to all other points. Scores are calculated by projecting points on the axis between endpoints. Select from the plot options. Next, select the methodology for calculating distances for the dissimilarity matrix. Click Update to apply your changes. Next, customize the graph. Select to display ellipses, variable arrows, or sample names. Next, select the grouping variable. Finally, select a color palette. Update to apply your changes. Scroll down to view the customized Bray-Curtis plot. Download the related data files and export the plot as needed. Switch over to the Scree-Plot tab. Select which plot options to apply. Select the plot type, either a variance plot showing both accumulated and individual variance, or a classic plot displaying eigenvalues. Select whether to display percent labels. Update the plot to apply your changes. Scroll down to view the plot. Download the results files and export the plot as needed. Access all data files from this analysis session through the Download option in the left-hand menu. Click the Results banner to open a drop-down menu of all data files. A list of all downloadable data files from this analysis session will open. Click on any file to download. The download.zip file contains all listed files. Download the WEGAN Docker file to recreate the WEGAN coding environment. Download the R source file to get the exact R script used during your analysis session. When done with exporting, click Exit to close the current module and return to the home page. We hope you found this tutorial on the Ordination module helpful.